A very good evening to all of you. Hope all of you are doing well and keeping yourself and your immune system healthy. So, welcome to the first webinar of Biotechnology Department of JIS University. The topic I will cover today is some basic part of immunology, which we regularly taught in our regular classes of BSc and MSc students. So, to start with immunology, we need to know the immune system first. And before moving further, please spare some time to appreciate these guys. These are all your immune cells which are actually protecting your body and keeping a constant surveillance 24 into 7. Okay, so they are the real hero of your immune system. So surely they are our friend who are protecting us, recognizing our enemies, and don't cause any harm to us to memorize all the things. হ্যাঁ বলুন হয়ে গেছে স্টার্ট and memorize all our things that is happening in our body. So, first of all, the things we should know before moving further is what is immune system? The immune system is actually the cells and all the tissue of your body, that is from your mucosal tissue, your epithelial tissue, and all the molecules those tissues and the cells are secreting. Those cumulatively form your immune system. Next, the study of your immune system is actually called the immunology. So it is a broad subject, a huge one, and everything is interconnected. All the cells, all the tissue, all the molecules they are very much interconnected to each other. And what is immunity? Immunity is when you are eliciting some resistance as a host to some pathogen and their toxic effects to prevent some disease which they are causing. So that is immunity. And this collective and coordinated response that you are introducing towards a foreign particle. A foreign particle is those particles which is not coming from your body, which is not self. Self means which is not mine. Everything in the immunology is based on the self and non-self recognition. Anything is not mine is non-self. So we have to elicit immune response against that non-self particle. So the immune system broadly is made up of two things. One is innate immunity. Innate means it is inborn. It is the first line of defense. Why it is first line of defense? Because when a pathogen or a disease causing particle is entering your body, the first thing that will cause the barricade against that pathogen is called the innate immunity. And when it fails, then comes the acquired or adaptive immunity, which is very specific, to a pathogen or a disease causing particle. So it will take some time to elicit an adaptive immune response. So the basic difference between the innate immunity and the adaptive immunity is the innate immunity is the first line of defense and adaptive immunity is second line of defense, we can say. And the innate immunity is inborn and adaptive immunity will acquire based on the pathogen or the particle your body is encountering. Okay, and last, innate immunity is not specific. For each and every pathogen, for each and every disease, all the cells or all the tissues of your innate immunity will elicit the response in same way. So it is very much non-specific. Okay. On the other hand, the adaptive immunity are very much specific. Okay. So the innate immunity is made up of all my anatomic barriers like skin or mucous membrane, your hair, your cilia, everything. The ciliated epithelium in your nose, your eye, your nails, etc., all are under the innate immunity. And what is the physiological barrier? Your temperature, your body temperature. That body temperature is somewhat not beneficial for many pathogens. And your pH, like your stomach pH, which is very much acidic. That acidic pH is detrimental for some pathogen. So they can't live there. If they enter to your stomach or interact with the temperature or get trapped in your mucous membrane, they will be deleted from your system. So these are very, very initial barrier. Those act in your innate immunity. And now comes the cells. Those cells are somewhat very much big eater. Big eater means I have written here that phagocytic barrier. Phago means eat, and cytic means cells. So cell eater. There are some cells which eat the pathogen. Okay. And inflammatory barriers, when you have cut, you got redness, swelling, pain, those are actually cumulatively called inflammation. So all these barriers, anatomic, physiological, your cellular barrier, your inflammatory barrier, all creates your innate immunity. Now, these are the anatomic and physiological barriers. Your lysozyme in tears that can lyse the many bacterial cells. The nasopharynx can remove the particle by its ciliated epithelium. The mucous lining of your trachea where the pathogen can trap and can delete it. Your physical barrier, your normal flora in your intestine like lactobacillus or streptococcus, they can actually create some barrier against the new pathogen. And always your stomach pH that is very much acidic. So what are the cells of the immunity? Now we will talk about some cells which act in the innate immune system. First of all, very important, which we see in our blood result when you got blood routine examination, like neutrophil is this to this percent, basophil is this to this percent, etc. So neutrophil is made up of 70 to 80% of your total granulocyte. So it is the first cell. When the pathogen breaches your anatomic and physiological barrier. So listen, when a pathogen breaches your anatomic and physiological barrier, it interacts with the cell of your immune system. So it is the first cell, that is the neutrophil, that will go to the site of infection, and it is phagocytic. So it can kill cells by eating them. It will create a phagosome and degrade that pathogen. And it has some big granules, like this type of granules. These are the granules. These all granules are filled up of myeloperoxidases, defensins, and some serine proteases. Those actually lysosomal granules that secrete upon infection and cause an immune response against a certain pathogen. But again, these are very non-specific. For all the pathogen, it will do the same thing. Now, eosinophil. Eosinophil, it fights against the multicellular parasites and some bacteria. It is not phagocytic actually, and it has a big role in allergic response. Eosinophil is a very low percentage, persists in a very low percentage. It has a role in inflammation, in parasitic infection and allergic reaction. Mast cell, it releases the histamine. They have large granules, and those large granules that releases histamine, that histamine cause the anti-inflammatory response. And mast cell also, like eosinophil, cause the allergic response, and it has role in microbial defense. Now, this is a very important cell in case of innate immunity or in case of adaptive immunity. Monocyte that circulate in blood. Okay. So whenever there is an infection, the monocyte from blood migrate to the site of the infection, and there, upon interaction with the pathogen, it differentiated the macrophage. Macrophage is a large cell eater, means they are large phagocytic cells. So they can't circulate in the blood. Actually, the monocyte, their precursor, they circulate in the blood. And when there is an infection, they travel to the site of infection and differentiated to macrophage. That macrophage is actually resident of tissues. And they are one type of antigen presenting cells. I will talk about it later. They can kill pathogen by generation of phagosome, and into this they can create ROS. ROS means reactive oxygen species. The reactive oxygen species like superoxide dismutase, hydrogen peroxide, they are in certain level are beneficial for cellular metabolism and signaling. But whenever the ROS level increase than the threshold level, it is very much detrimental for a certain cell. So they create a very much increasing level of ROS in those cells and kill those pathogen, and also release some lysosomal enzymes that will kill the pathogen. This is a very important cell, which is called dendritic cell. Dendritic cell is actually, dendrite means the branch. If you see that it has a branched structure, it is very much important in your immune system because it acts as a professional antigen presenting cell, because it is one type of cell which can engulf pathogen through phagocytosis and present the antigen to certain cell. Which cell? I will come later. Yes, it is phagocytic. Now, this is the natural killer cell or NK cell. As the name suggests, it can naturally kill other cells. Which cells? The cells which are non-self. Means it is not very specific, okay, but they kill any virally infected cells and other transformed cells in our body like tumor cells. Okay, so those natural killer cells or NK cells can kill tumor cells and viral infected cells via process called apoptosis, which is a programmed cell death, and cellular lysis. They can release some mediators which can cause cellular lysis called perforin or granzyme. Now, your pathogen, suppose, breaches your physiological and anatomical barrier. Whenever they breaches your physiological anatomical barrier, the next thing in the innate immunity, that part, in which these cells participate is called inflammation or inflammatory response. It is actually a result of the activation of all innate immune cells and their effector function. So the innate immune response is initiated within an hour. Okay, when you are infected by a pathogen or you have a wound. If you have a wound, then bacteria or virus or any other pathogen have free entry into your body. So they can easily breach your physiological and anatomical barrier and can directly interact with your cell. So your tissue is damaged, your a bacteria or virus is infecting you. So what next? Mast cell, they release histamine. Okay, when you have a wound, that cause a increased blood flow. Why increased blood flow? Because it will take all the cells that is circulating in your blood to the site of infection, but they are in blood and the pathogen is in your tissue. So we need something to move those cells from the blood capillary to the site of infection in the tissue. So first of all, our blood capillary widen. Okay. This widening is incorporated with increased permeability. This blood capillary widening and increased permeability of your blood capillary cause the heat and redness of that site. It is actually you are healing, your cells are acting. Okay, your inflammation means your cells are acting. Okay, and as the capillary widen, your capillary has increased permeability, all the proteins and fluids in your blood, they just move to the site of infection, that is in your tissue. So as those fluid and proteins are migrating continuously to the site of infection in your tissue, it will cause swelling. Okay, that will fill with pus. ঠিক আছে, that is the dead cell pus. Actually ওখানে যেগুলো the cells that is actually uh, creating inflammation, and they are continuously causing a debris, they are actually causing the pus. Now this the first cell from the blood that will move through the pore of the capillary and the endothelial cells of the capillary is the neutrophil. The neutrophil and also macrophage, those actually interact with the pathogen engulfed by phagocytosis and release some chemokine. Chemokine are the substance which actually act as a chemoattractant, which attract other cells like basophils, mast cells, eosinophils to the site of infection. And ultimately, they are actually healing the site and your blood at that place being clot so that the infection cannot spread. Okay, and the systematic response is the fever and proliferation of leukocyte that will cause you pain. The fever is actually means that your body is healing through our inflammatory response. Now this is the innate immunity and inflammatory response. So after that, if all those cells fail to elicit a successful immune response against that pathogen, means it is not your system is not free of that specific pathogen, then the adaptive immunity will start. The adaptive immunity, as I have told you, that it is very specific, it is diverse, it has memory. Memory means today, if I am encountering a pathogen after 6 months or 12 months, I will recognize again that pathogen and elicit an immune response. Okay, it will not take time. So the first response, the first adaptive immune response against a new pathogen or against a new infection take some time, 7 to 10 days, actually 7 days. So humoral immunity has two arms. One is humoral immune response. Humoral came from the word humor. Humor means body fluid. Actually, the body fluid, in our body fluid, there are some protein which is called antibodies, which recognizes our foreign particle, which is called antigen, and elicit immune response against them. So those antibodies are produced by B lymphocyte, and there is cell-mediated immune response, which is elicited by T lymphocyte. So the very specific role or the main role of B lymphocyte is to produce antibodies, and the role of T lymphocyte is the direct killing of infected cells and the release of some mediators or protein mediators which can elicit signaling response in other cells called cytokines. And the T lymphocyte also help other cells to activate them. So this is the innate immunity and adaptive immunity when they start. The innate immunity is made up of basophil, eosinophil, neutrophil, mast cell, and natural killer cell. The innate immunity can persist up to 72 hours. That is 3 days, 0 to 72 hours. After that, there will be a 4 to 5 days window when the antigen will be presented to the adaptive immunity, to the cells of the adaptive immunity, that is the T cell, okay, via antigen presenting cells. So the innate immunity, the first line of defense, which starts at 0 hours and persists up to 72 hours. After that, all those pathogen will move to the adaptive immune cells via some antigen presenting cells. So the antigen presenting cells is actually act as a bridge between the innate immune response and adaptive immune response. Without the antigen presenting cell, the adaptive immune response won't start. Okay. So this is the function of B cell, as I have told, that the function of B cell is to produce antibody and is to produce memory B cell. Memory B cell money. What is the meaning of memory B cell? Memory B cell is those cells which actually not participating in the primary infection. Okay. When there is a secondary infection, they will go to that site and produce antibody against that particular antigen. And so what is antigen? Antigen is the foreign or either particulate, that is can be cell, bacteria, virus, or any part of their cell, means any protein particle, any polysaccharide or a lipid particle of them are termed as antigens. So they are non-self. Non-self means they are not mine. Okay. Anything not coming from the cell will be recognized as antigen by your immune system, and they will elicit immune response against them. So the antibody, they are the protein We called it gamma globular protein. That is actually a Y-shaped protein. It has a Y-shaped structure. It present in serum, and they are often called immunoglobulin. It is the gamma globulin protein, and they are from the immune response. So immunoglobulin protein. Now, the antibody can be used in many serological tests. Serological tests means if you have an infection, the antibody response within your body will rise. Okay, so the first antibody that will elicit the response against a pathogen is called IgM. So IgM is the primary immunoglobulin that released by the activated B cell against any antigen. So we can say when there is a very new infection or just a asymptomatic infection or a primary infection, then the IgM is the major antibody we can found in any individual serum or plasma. After that, when after some window period, when there is an acute infection or chronic infection, so the infection is old, then the IgM class will convert to IgG. They have the basic difference in their chain. I will not go there. Okay, so IgM is the primary antibody against the primary infection, and again in the secondary antibody we can found the IgG. So based on this, we can create a rapid antibody test. So how this rapid antibody test carry out? So you can collect blood, serum or plasma from any suspected individual or any infected individual. Okay. You can see the capillary. This is the capillary tube. We call it a capillary tube. So in this capillary tube, there is labeled antigen. Okay, the antigen you are suspecting that against which an individual is eliciting immune response. So they have high level of IgM or IgG in their blood, in their body. So that antigen that chemically bind with IgM or IgG in the capillary, and so they form the antigen antibody complex. Now, what you will do, you just collect 2 to 3 drops of fresh blood or serum or plasma from a finger prick and just place 2 to 3 drops, and with a buffer, as it is a lateral capillary action. So for the capillary movement, you need a buffer. And this allows the diluted sample to move through the capillary tube by capillary action. Now, after the reaction, the antigen antibody product that passes over anti-IgM and anti-IgG antibodies. So first of all, your capillary has labeled antigen. You have maybe IgM or IgG in your blood. You just put 2 to 3 drops of that blood and place and put some buffer. And when there is a capillary action, your immunoglobulin or your antibody, that can be IgG or IgM, will react with the specific antigen, and through the capillary action they will move. After that, they will interact with another antibody which is anti-IgM, that is antibody against the IgM molecule, and antibody against the IgG molecule. So they will capture that IgM and IgG that is bound with the specific antigen, And as those antigens are labeled and the buffer has some substance, they will give you a red type of red colored band or a red line. And there is a control line in which there is no antigen, only the specific antibody is present there. So if there is a faulty experiment or your kit is faulty, the control line, you will not see any control line there. So if there is no control line, your data is invalid. So you can see that when there is a positive result, you have two line, that is control and in IgG, control or in IgM, and control along with IgG and IgM both. So they have, so only when have IgM only, so then the antibody has been detected and the result is positive for the IgM antibody. Same as IgG, the result is positive and the result is positive for the IgG antibody. And the IgG and IgM, for both, they have positive result, means that the infection was there, okay, but now the individual is actually getting rid of that pathogen or getting rid of that symptoms. So moving on to the next slide. Now there are various T cell in our adaptive immune response, which is called T helper cell and cytotoxic T cell. The T helper cell has different subtype, which is called T helper 1, T helper 2, T helper 17 cell, and T follicular helper cell. So coming to the cytotoxic T cell, cyto means cell, toxic means it is lethal to a cell. So this type of T cell, that is the cytotoxic T cell, can directly kill virally infected cell and also cancerous cell. The T helper cell, as the name suggests, they actually help other cell. To which cell they render help? Those are macrophage, other T cell, that is cytotoxic T cell, and B cell. The T helper cell, actually they cause the activation of macrophage and B cell. So how they help the other cell, that is specifically the T helper cell. The T helper cell helps the other cell by producing or secreting cytokines. The cytokines are the proteins which secreted by the T cells and many other immune cells that will act in three different way. That is in autocrine action, that is it acts on a same cell from which it is secreted. The paracrine function, that is it is effective to the nearby cell. And the endocrine action, that is they released from a certain cell and they circulate in your blood system or the lymphatic system, and then they are affect the distant cell. So this three way, these cytokines can help the other cells. So T cells, basically, specifically T helper cell, they produce these cytokines to help other cells. And as I told that these are the antigen presenting cell. These antigen presenting cells are called dendritic cells, macrophage, and B lymphocyte. All three cells can process and present antigen. There are some pathway to processing of antigen. So this is a very broad subject, and they can present the antigen. So without these cells, the T cell cannot recognize a specific antigen. Again, those cells possess a protein on their surface via which they present the antigen to T cell, and that protein is called MHC, major histocompatibility complex. In uh, in conventional way, all the nucleated cell, they possess MHC class 1 on their surface, and all the antigen presenting cells, they possess MHC class 2 on their surface. So this dendritic cell is the link between adaptive immune response. As I have said, that those antigen presenting cells are actually act as a bridge between the innate immunity and the adaptive immunity. Without them, the T cell cannot act. So how a T cell is activated by a antigen presenting cell? The major or the professional antigen presenting cells are the dendritic cells. Remember these dendritic cell, this is the dendritic cell, the branched structure. So these dendritic cells are actually act as antigen presenting cell or professional antigen presenting cell. So they give T cell three different signals. First signal, they present the antigen on their surface via MHC2 protein or receptor, which is recognized by the T cell receptor. The TCR means T cell receptor, which is expressed on the T cell. Next, the signal 2. The signal 2 is the co-stimulatory signal. The antigen presenting cell or the professional dendritic cell, they express the co-stimulatory molecule that is called CD80, that is cluster of differentiation 80, and cluster of differentiation 86. They have other names like B7.1 and B7.2. Cumulatively or collectively they called as B7 molecule. So the co-stimulatory molecule actually B7 molecule that is expressed on the antigen presenting cell, that is dendritic cell. And those co-stimulatory molecule are recognized by the CD28 receptor that is expressed on the T cell receptor. So the first signal, that is the interaction between the MHC and T cell receptor and antigen, that gives T cell the signal of activation. It activates a specific T cell. Before that, the T cell is matured but not activated. The second signal, that is the co-stimulatory signal between the B7 molecule and CD28, it gives the T cell the signal of survival and proliferation. So after activation, the T cell will proliferate. Proliferate means they will increase their number. They increase their number to help other cell. Some cells of that clone, of that specific T cell clone, will help other cell, and some will create a pool of T cell. That pool of T cell won't take part in that immune response, but they will be in a storage. They are called memory T cells. Okay. Next, signal 3. After signal 1 and signal 2, there is another signal which is given by dendritic cell in the form of cytokines. That cytokine will act on T cell specific receptor for the differentiation of the T cell. Differentiation means this T cell is now activated and proliferative. But how we can make the different subtype of T cell like T helper 1, T helper 2, T follicular helper cell, how? From this step, this is the signal 3, that is the cytokine. Based on the cytokine signal or the different cytokine signal, the T cell will give rise to different subtype of T helper cell. that is called T helper 1, T helper 2, and T follicular helper cell. So after activation, those T cell, they can activate the B cell. So first, the T cell need to be activated by the antigen presenting cell. So coming from the innate immunity, just to brush up the memory. So a pathogen breaches your anatomic barrier, your physiological barrier, get inflammation. After that, after 3 to 4 days, your adaptive immunity just started, because your antigen presenting cell, that is the dendritic cell, they take that antigen from the innate immunity part and migrated to the adaptive immunity part. That is the what is the site of adaptive immunity is actually the lymph node and secondary lymphoid organs. They are the sites of adaptive immunity, where the antigen presenting cell, they present the antigen to the specific T cell. And then the T cell is activated, proliferated, and differentiated. And after differentiation and activated, they can activate other cell as they are T helper cell. Their role is to help which cell? To B cell, so that the B cell can produce antibody. Okay, and B cell can proliferate to create a pool of B cell, that is the memory cell, and to produce antibody. The antibody producing B cell, which is called plasma cell. So that T cell can activate B cell, can produce memory B cell, and can produce antibody producing B cell, which is the plasma cell. The T helper cell, also the macrophage, needs help from the T cell for the macrophage activation. In the innate immunity, the macrophage is actually engulfing the cells, but when they can't do anything, then they need help from the T cell, that is from the adaptive immune response, so that the macrophage being more activated and can stimulate microbial defense. Those are the big substances in their cell. Those are filled with antimicrobial substances. So T cell, that activated T cell, can help the B cell and the macrophage for their increase in effective function. So those are the role of T helper cell. Also, the cytotoxic T cell needs activation from the antigen presenting cell. It is not that only T helper cell needs antigen presenting cell for their activation. Also, cytotoxic T cell needs activation from the antigen presenting cell. So the cytotoxic T cell, they directly kill the infected cell. What type of infected cell? The virally infected cell and the tumor cells. They release some granules called perforin, granzyme, said glycine. Those actually create pores in the membrane of the infected cell, and through some systematic cellular signaling, they cause apoptosis in that cell. Apoptosis means programmed cell death. The cell will form apoptosome. Their DNA will be fragmented. There will be cellular blebbing, chromosome condensation, and cell will die eventually. So cytotoxic T cell, they can directly kill infected cell. T helper cell, they can help other cells via releasing cytokine. So the basic question that everybody asks, that how come then the tumor cells cannot be recognized by your immune system, right? So if our immune system is so much equipped with all those things, why the tumor cells can evade this immune response? So actually our immune system or our immune cell recognize tumor cell. they can recognize tumor cell in first instance. Okay, when because if you think that every hour or every minute there is an abnormality in our system which can cause cancer. Okay, the cellular division, mutation which can cause cancer. We are also exposed to various environmental factors which can cause cancer. So why but we are all the time there is no cancer in our system. Why? Because the immune system is continuously recognizes those cancerous cell and eliciting immune response against them. When they can't, when the cellular response or when the tumor cell or the cancerous cell are aggravating their response, when they actually lost their expression on their surface, their metastasis is aggravating, there are increase in the cellular proliferation, so that in that case your immune system are overdoing their work, but that is not sufficient to defend your body from that overly growing cells. So there that our immune response fails. Okay, so the tumor cell can avoid the immune recognition by two broad response. One, they can lose the tumor antigen expression on their surface and with very low expression of MHC1. If they have high expression of MHC1, then the CD8, that is the cytotoxic T cell, can recognize them. So they lower their MHC1 expression on their surface. On the other hand, the specific tumor antigen that can be recognized by the tumor response as non-self, they just lost their expression on their surface. Okay, and number two is the initiation of the tumor immune tolerant tumor microenvironment. Tumor microenvironment means the environment just in the proximal vicinity of the tumor cells. So the tumor cells that release various cytokine and other factors like IL-10, that is the cytokine, transforming growth factor beta, that is prostaglandin E2, and vascular endothelial growth factor. Also, they can express some checkpoint on their surface. The specific checkpoint I will talk about, that is program death ligand 1 and 2 and cytotoxic T cell mediated antigen, that is CTLA4 expression. And also they can recruit the tumor associated macrophage and myeloid derived suppressor cells. The tumor associated macrophage is actually have suppressive function against the immune response. So they can suppress the immune response, that tumor associated macrophage. MDSCs, that is the myeloid derived suppressor cells. They are myeloid cells, but not a conventional myeloid cells. They have suppressive functions. And regulatory T cell, they can actually suppress the T cell activation. So how we can get rid all of those? So this is a very broad subject again. So just one thing, we can do immunotherapy. So the CTLA4 and PD1, I have showed here only these two. This pathway can be blocked by a specific antibody called monoclonal antibody. Okay, so CTLA4 blockade allows for activation and proliferation and more T cell clones, and also they can reduce the regulatory T cell mediated immunosuppression. So the regulatory T cell also also express some CTLA4 on their surface, so they can suppress the T cell response. So if we use the monoclonal antibody against CTLA4, so that can be reduced. And PD-1 pathway blockade, that restores the activity of the anti-tumor pathway. Actually, the PD-1 receptor is expressed by the T cell, and the PD ligand 1 is expressed by the tumor cell. So various types of tumor cell can express program death ligand 1 on their surface, and they can be recognized by the T cell, and the T cell activation will be inhibited. So if we can block the PD-1 receptor by anti-PD-1 antibody, so the T cell will be activated and they can recognize the tumor cell and elicit immune response against them. So these two drug, there are so many monoclonal antibodies there, like 5 or 6 in these two category, that is anti-CTLA4 monoclonal antibody, that is lipilimumab and tremelimumab. They are actually in phase 3 trial, I guess, against the lung cancer and melanoma, metastatic melanoma, and also the monoclonal antibody against PD-1, that is nivolumab and pedilizumab. When there is anything in the last that is called termed as MAB, means monoclonal antibody. So rituximab, then trastuzumab, those are actually monoclonal antibody. Now, here your innate immune response is actually inborn, but you can acquire, okay, through lifelong process and through vaccination. So anything that will boost your immune response, anything that will boost your immune response is called immunization. Means you are being immune to a certain disease or certain pathogen. So immunization can be classified into broad categories. One is called active immunization, another is called passive immunization. And both have two categories called naturally active immunization and acquired active immunization. What is naturally active immunization? When you are exposed to various antigen in your lifetime, okay, so today you are exposed to 5 antigen, tomorrow we will be exposed to 2 antigen, and you are not getting sick. So your body is actually fighting against that specific pathogen or antigen. So your immune system is actually evolving. So you are immunized against that specific antigen. So naturally you are gaining immunity against that antigen. What is acquired? When you are getting antigen directly in your body in a suspension or in a biological preparation. So any antigen as a whole cell antigen or any protein like tetanus toxoid. So this is the acquired immunization. Polio vaccine, this is the acquired immunization. Then rubella vaccine, or MMR, means mumps, measles, rubella vaccine. Then DTP, diphtheria, tetanus, and pertussis vaccine. Those are actually acquired immunization that came through the vaccination. And what is passive? Passive is actually you are not exposed to any antigen in this immunization. What you are getting antibody in a natural way. How you can get antibody in a natural way? Obviously from your mother. When you are fetus, then your antibody you can get from your mother through placenta. The antibody from your mother will flow through the placenta and get into your body. So this is the naturally passive immunization. And also during breastfeeding, you can get the antibody from your mother. And what is acquired passive immunity? When you are getting preformed antibody from an individual, say, I I just came to know yesterday that CSIR, ISCB, and Belaghat ID Hospital, they are actually going to do passive immunization. Passive immunization means the COVID-19, the patient who actually survived the COVID-19 infection, they actually formed some antibody in their system. So the thing is, researcher will take the serum or plasma from their blood, and that will administered in the patient body. So it can give immune response, it can elicit immune response as a antibody against that specific antigen, that is coronavirus. So this is it. Thank you for being there. Thank you for listening. So this is the antigen presenting cell. This is the actual scenario. So the antigen presenting cell that showed the antigen to that helper T cell, and helper T cell should be thankful that it can now elicit the adaptive immune response and help the other cells to create a specific and very much specific immune response against certain pathogen. Thank you all for listening. So I'm ready to take any question now. So if there is any question, the session is open for questioning. Thank you, sir, Manibroto sir, and thank you all the students, Lisa Ghosh, Prokriti Paul, Kulani. Thank you, madam. Thank, thank you very much, Dia, Jilam sir, Mamta. Okay, good evening to you all. So if there is no question right now, so I think I can sign out.